Thank you so much. Well, first, thank you for this really kind invitation. It's a thrill to be here with this broad audience. Um, but before I get going, I'd really like to thank the organizers, not just for having me, but over the past few years, the current organizers and the previous ones for actually running the series. I think this has been absolutely invaluable to the community, during COVID times and beyond. And I really like to express my appreciation there. This has been fantastic. So the more joyous I'm here. So in this kind of first, less than half hour, I'd like to give a brief intro into stochastic agent-based Monte Carlo simulations, most of which I think you know how that works. So I'll be focusing rather on a few, minor things that you need to worry about. So I'll talk a little bit about chemical kinetics, which is my kind of general language for this, but that also applies to epidemics and population dynamics and what have you, and emphasize where fluctuation and correlation effects are important. Hence, we are actually doing these individual-based simulations. And I'll give a few tips and caveats and examples. I'll be rather short there because that'll be featuring in the following research talk as well. And I'd like to advertise this very recent AJP paper that we wrote, which is meant to be introductory for, hopefully beginning graduate students, advanced undergraduate students, to explain how that works, and also for advisors who may try to learn about this. And again, as opposed to kind of typical research publications, it does talk about some things that can go wrong and how to avoid that, and some subtleties that are never mentioned in the actual research papers because the wrong stuff we don't tend to publish. Okay, so this paper was done with Mohamed Swailim, Uli Dobrmüsel, and Ruslan Mukhamadyarov, all former students of mine. And then there's some other references, on population dynamics and my textbook. Okay, so chemical kinetics is really about some interactions, physical or otherwise, that alter the constituents' identity. So this is, of course, when you have transformations in biological systems, when you have biochemical reactions, nuclear reactions, particle physics, but also ecology, epidemiology, when you go from susceptible to infected, for example. And just to make this concrete, let's look at this binary reaction. So we have two different species, A and B, going forward with some rate lambda, going backwards with a rate sigma. So I'm going to treat this as a stochastic process. These are random events, just because fundamentally maybe there's quantum mechanics, but also because there may be all kinds of random external influences. So I'm treating these as a stochastic process. And that means we're going to write down a master equation. Maybe first to describe this, I'd like to emphasize what A plus B going to something really means, which is really encoded in this language, is that these things have to find each other. They have to be at the same space-time point. So they need to be spatially and temporally correlated. It's not kind of A's and B's are independent. The backwards process isn't. That just happens spontaneously. So here's the master equation. That's a time-dependent evolution equation, classical, for the density or number of A, B, and C constituents. And there's gain and loss terms, and that's just the balance. In the loss term, we are starting with Na particles of type A and B particles of type B, and so on. And so when we have both of them present, this forward reaction can happen. The backward reaction can happen when we have NC particles present. And we can pick each of these, so we get these kind of stoichiometric, if you like, coefficients. And then the gain processes are similarly. For the forward reaction, if we want to end up at Na and B and NC particles, we have to start initially with one more A, one more B particle, and one less C particle. So we need to start with that initial configurational probability, multiply with this numbers and the rate. And for the backward process, we have the backward rate, and we need to have one fewer A, one fewer B, and one more C particle to end up through this process at Na and B and NC. So that's your master equation gain loss balance. Based on that, you can just look at the time evolution for averages. You can just look at the average concentration of A, B, and C particles, indicated with X here. And all you do is you take the time-dependent configuration probability, multiply with that number, sum over all configurations. That's definition of an expectation value. And when you go through this, inserting this one here, play with the summation indices, which carry all possible labels, you can readily derive that the AB density number goes down while the C number goes up. Of course, this is what is encoded here. And then you get two terms here. For the backward reaction, it's just proportional to the C number. That is what you would have, quite obviously, it's just a linear process. but the forward direction is really proportional to the second moment, simultaneous second moment of having both A and B particles at the same, essentially, place. And so that is precisely where, when you do what is called a mean field rate equation, you make a huge assumption. You're basically factorizing this into the product of the A and B densities. And that is what I'd like to emphasize, because in any kind of typical math biology or physical chemistry textbook, this is where you start. But that means you're already starting at a fairly severe approximation, which can be justified, but it needs to be checked. And so this will work. You can kind of forget the difference of this, which is actually the correlations. You can forget that when you keep mixing your system. So you're kind of destroying any kind of correlations, make them really, short range, then you might be getting away with this. That typically tends to work sometimes if you have really large population numbers, large number of species, you might get away with that too, because the relative fluctuations, might be small, scaling down like 1 over square root of n. But that needs to be checked. And I'll show you counter examples where the very kinetics that your reactions describe actually induces correlations, even if you don't put them in and they're not externally enforced, they're just kind of self-generated through the very kinetics. All right, so, but if you do this approximation, you end up at these coupled nonlinear rate equations, and they become nonlinear precisely because you're doing this factorization. Up to there, everything for the master equation for the configuration probabilities is linear. You get an infinite hierarchy of linear equations for infinitely many of these, increasing correlations or moments. But as you do this factorization, you get a much reduced set of ordinary differential equations, which have become nonlinear because of the approximation that you're using. So I've already emphasized these bits here, and I'm going to show a few places where this is prominently showing up, also in the context, of course, of biological systems. And so you can have this kind of dynamical correlations generated by the very kinetics. You can have spontaneous formation of noise-induced structures, which is, of course, vital in biological systems. So when that happens, you need to really be careful about these mean field equations. And by the way, also kind of mean field reaction-diffusion equations. Just adding spatial components and looking at local coarse-grained density still entails this factorization. So you haven't actually solved the full process either. And so computational, and of course there are critical fluctuations, and you have to worry about systems with a small number of constituents. So if you're close to any kind of extinction or fixation, you need to worry especially about this stuff, because relative fluctuations become large. So computationally, you go about this with agent-based simulations, which is what I'm going to be talking about. But then there's also a few analytical approaches that I should at least mention, because I've also done some of these. Mostly in one dimension, you can do certain exact systems, A plus A to 0, you can do exactly, and there's a few others. And those are fantastic because they give you benchmark solutions. Of course, you cannot typically generalize that to anything as complex that you might need in biology, but you're still learning fundamentally what can be going on. Beyond that first brutal factorization I showed you on the previous slide, there's more refined mean field theories. You can do higher order correlations that you kind of break up. There's dynamic cluster expansions and the like. And depending on your problem, some of these work nicely. And then there's the systematic approach where you map the stochastic master equation into a path integral representation. And then you can play with that. And of course, you cannot solve that exactly either, but then you can do systematic perturbation theory and the like, and then map that back to stochastic PDEs and have some fun with that, which we did, but I'm not going to talk about that today. All right, so here's some prominent examples for the relevance of fluctuations and correlations or kinetics-induced spatial correlations. And of course, these are for toy models first, right? So these are meant to be in a kind of typical physics approach to any kind of more complex systems. We look at very isolated, simple systems, try to figure out what we need to maybe worry about, and then, maybe utilize that later where needed. So the first one is pair annihilation or coagulation of particles or species, individuals of the same species. So we have A plus A coming together and either annihilate or just coagulate to the same particle. When that happens in your low dimensions, I'll tell you in a second what low dimension means here. And these things have to find each other, say through nearest neighbor hopping or continuum diffusively. You have a diffusion limitation when the density becomes low. So when you have a large, number of particles together, they always find a neighbor, and then you essentially just have a reaction limitation. So the reaction rate limits, how fast the process will progress, and that's essentially governed by the mean field rate equation. That's fine because we have a large number of species. But as we go to low numbers, the different A particles have to find each other, like in this kind of diffusive sketch here that was actually analyzed, over 100 years ago by Mario and Smoluchowski. And there you notice that, what can happen is as I'm getting closer, I'm going to find all other particles that are close, and the nearby neighbors will just annihilate. And so you're left with kind of just through the very kinetics of this process, you're left with survivors that are increasingly far apart from each other and actually spatially anti-correlated. So this reaction process, just by looking at the survivors, induces effective anti-correlations. So it's almost like fermions, and actually in one dimension, you can map that to free fermions. And so you get depletion zones. As you analyze this in somewhat more detail, you find that, if you're in more than 2 dimensions, your random walk is very nicely mixing. It's going through all space, and it's just a very diffusive process, avoids or quickly destroys these depletion zones. But in 2 dimensions and less, these depletion zones are persistent, essentially because of the recurrence probability that random walks have in 1 dimension and 2. And that actually changes the kind of mean field description. If I write down the rate equation for this one, straightforward exercise, you find that you get a 1/time temporal decay for the particle number or density. Once you take into account this kind of diffusion limitation in 1 dimension, it's 1/square root of time, so much slower. And so this is not just kind of a prefactor quantitative correction, it's a massive change in the power law, which of course, try to fit to, you get arbitrarily wrong answers. And in 2 dimensions, really in the sense of a critical dimension of critical phenomena, you get a logarithmic correction. One other way you can describe this is in terms of a renormalized effective coarse-grained reaction rate, which really just as the density decreases, goes down. And likewise, as time goes on, the reaction rate goes down. And that's again effectively taking into account that you need to overcome these anti-correlations and the increasing typical distance between these particles. And that has been nicely demonstrated experimentally in this, 10 years, 13 years ago in this physics letter by Allen and others, where they looked at the quenching of excitons in carbon nanotubes, quasi-1-dimensional systems. And they very nicely see this crossover from reaction limited 1/T decay to diffusion limited with normalized reaction rate 1/square root of T decay. So this is real. Of course, this is a bit of an arbitrary, not arbitrary, artificial system. Does it show up in simpler systems? Yes. You do ligand binding, things like that. You have to worry about similar kind of anti-correlation effects. And we've done that. So different but related is distinct species. So now we have 2 species, A plus B, that have to find each other and then say annihilate. That superficially looks very similar, and indeed the mean field theory also looks similar. However, the difference not stated here is reactions only happen between unalike species. There's nothing that the A's do to each other, there's nothing that the B's do to each other. So as you let that go, you have close neighbors of A and B annihilate, and you get inert zones of A's and B's. So here's the simulation word line picture where we're going in 1 dimension, that's X, down in time. And we see if we just start randomly, in this case, we had a majority species of blue guys. The red ones will find some blue ones, annihilate, and then you're left with kind of large intervals of blue, smaller intervals of red, large intervals of blue. So you get species segregation, which affects what's going on. And that is especially drastic if the particle densities are equal. If they're not equal, you get a stretched exponential decay. If they are equal, you get a power law reduction from 1/T in mean field to 1/4th root of T in 1 dimension, 1/square root of T in 2 dimensions. And you get this kind of reduced power law, which induces much slower decay kinetics in any dimension less than 4. But aside from that slow decay, what is perhaps even more important, I think, for biological systems is that this allows the species segregation and thereby spontaneous formation of patterns, right? Where you have reaction fronts that separate A and B, and the A's and B are separate. And that had just happened spontaneously. Yeah. So that we found is very important in epidemic and population dynamics kinetics. And that being persistent less than 4 dimensions, I think is really crucial for biology, why we see spontaneously forming structures. And then lastly, the obvious one that everyone has been familiar with for decades is when you near a continuous, of course, phase transition, such as an active to absorbing or extinction threshold, you really have to worry about fluctuations. And so there's two typical cases. One is this kind of generic situation where you have particle production, or this coagulation I was talking about, and spontaneous death. The coagulation is really just kind of a limiting process that, in the active phase prevents the particle density to explode. So you have kind of finite carrying capacity, if you like. That one's called the simple epidemic process, and its critical dynamics is described by universal features of critical directed percolation, because this stuff written down in kind of now time going to the right and space perpendicular, you just draw these processes, say on a square lattice that you tilt, you just generate these directed percolation clusters, and then survival means you start here and you go through the entire system in time and there's still activity, while extinction means this ceases, and right at the critical point you get the scale invariant features, but with anisotropic scaling in the time and space directions. And then you can add debris, which is kind of shown in the SIR epidemic process. So that's a general epidemic process. In that case, you end up with dynamic isotropic percolation. So spatially, the clusters look like isotropic percolation clusters at the extinction threshold, but there's other fun dynamics. And then lastly, and I'd like to emphasize that too, you can be just in a finite system that is sufficiently small, depending on what your rates are, that when you have an absorbing state, you're actually finding it. So in mean field theory, in these rate equations, total extinction tends to be unstable. Whenever you add, a tiny fraction of particles, and that could be 10 to the minus 140, the system will expand and grow again just because you have a linear growth process typically. That is not applicable if you talk about particles with individual numbers. If you just don't have continuous densities, that can be arbitrarily small, but you just have 1, 2, or 3, and you have a finite system, and there's an absorbing state with probability 1, you're going to find it. Question is, how long does it take? And for a large system, the survival or the extinction time scales typically exponentially with system size. So if I go to sufficiently large systems in any kind of real experiment or real simulation, or even on Earth in kind of evolutionary timescales, I'm not going to see that extinction. But in small systems, I am. And then we get to these kind of finite system absorbing state transitions, stochastic extinction ones, which is definitely not captured ever by any kind of deterministic rate equations. All right, so that was my kind of general spiel on why we need to worry. And that takes us to these stochastic Monte Carlo simulations. And it's really just the implementation of these reactions on some kind of spatial system. It doesn't have to be spatial, but I'd like to do this on a spatial system. And so we could do this on a lattice. If you're worried about epidemics, you might really want to do this on a, near friend neighbor network. Whatever is most applicable to the system you have at hand doesn't really matter. And here I'm explaining this for the Lotko-Volterra predator-prey competition. So we have predator death, we have spontaneous prey reproduction, and a nonlinear process where we have this binary reaction again. Now A and B need to meet, and when they do, these prey particles B get replaced by a predator particle of some rate lambda. So that's different to the A plus B to 0, but it has the same flavor and it has the same kind of critical dimension for spontaneous pattern formation. And then say we have, in addition, used neighbor hopping rates, but that depends on what kind of restriction you put on your lattice or network node occupation. If you restrict that to say 1, then you need to generate new particles like in this prey birth process on adjacent sites, which automatically induces nearest neighbor hopping, so it automatically induces diffusive spreading. And if you don't have it, you can also implement it on top. So what you do in these simulations is very simple. You just throw out an initial configuration. Then you pick a particle at random of either species. And then if it's here's the example. If this is a prey particle, then you let it reproduce with some probability that you just set. and then you just throw a random number from your random number generator. And if that random number says less than that set reproduction probability, you let this thing go. If it's bigger than that, you don't. If you have a predator particle, that might just die away. Again, just decided by throwing a random number. Or you check if in its neighborhood there is a prey particle. If there is one, you throw a random number again. And if your constraint is satisfied, then you let this reaction happen. Otherwise, you don't. And then you just cycle. So this is standard Monte Carlo simulations. You just do a lot of these loops. Sometimes this is not effective, because if these rates are small, there's a lot of non-things happening. So you have a lot of unsuccessful trials. And then you call a time step, so we call Monte Carlo step. You call that when on average each of your present particles has had a chance to do something. So you don't go through all the particles, serially or really make sure all of them have happened, but you just randomly pick them. And so in this kind of random sequential process, you just want to make sure that on average each of them had a chance. That tells you, of course, that there's a lot of microscopic artifacts, right? I mean, there's a lot of choices you can make in which order you do these reactions and what have you. And I'll comment on that briefly. Now, also typically you do periodic boundary conditions, you let that go. And if you do this for that system here, you get these kind of spontaneous pattern formations. Again, essentially driven by these binary reactions. Few comments. First one is, what we're really doing here is we're trying to compute a sampling of the master equation configurational probabilities. So you have to do sufficiently many, samples to get effectively the time-dependent averages or correlations that are given by the master equation. But you're not actually directly simulating the master equation, you're just simulating the same processes. Second comment is there is a very, popular version of this that was originally called kinetic Monte Carlo, is now typically called Gillespie algorithm, where you do away with the kind of lost inefficiency of sometimes things not happening, and you always have a reaction happening. You just update the corresponding time exponentially. So you essentially say, if this is a Poisson process, then the probability for any reaction to happen is exponential in that rate. And so if it does happen, you just advance time exponentially. That's really important if your rates are very, very different. So you have a large timescale separation, because otherwise you just lose the rare processes. So for sampling of rare events, that's efficient. If, however, your rates are typically of the same order, it's quite inefficient, because what you have to instead do is, if you're really just interested in the stationary situation, fantastic. If you, however, want to actually probe the full-time evolution, you get this nonlinear time advance. And so if you really want to probe the full temporal evolution, you have to run a lot of realizations to get that nicely under control. And that tends to eliminate, any kind of advantages you might have had if the rates are on the same order. So we've always found just doing standard Monte Carlo is just as good and simpler, and you cannot make as much mistakes of, mapping it to some real time. Anyway, you need to do sufficiently many runs. Now, in this business, you don't know what that is. That depends on your problem. It depends on the size of your fluctuations. And if we knew that in advance, we wouldn't be running this, right? So that just means there's trial and error. And if I had a nice overall rule on telling you kind of what order you need, I don't. So you might, in some cases, get away with 100 simulations, but if you're near a critical point, you might need millions. And if you have a disordered system, perhaps more. So it's really depends on what kind of statistical errors you run for and what the intrinsic fluctuations look like. Then, as I already alluded to, there's a lot of algorithmic details. They really should not matter. If they do, they really should just affect the short time regime, which is not what you're asking about. I'm not asking about the time scale of an individual reaction. That really should be treated by quantum mechanics or something else, not by our kind of very effective model. So you're really asking for time scales much longer than this, and length scales much longer than your lattice size or your continuum, microscopic ultraviolet cutoff, whatever you have. If they do matter, you have to really question your model if they matter in the long time, because then you have a non-robust stochastic model. And if that's not robust, then modeling by itself becomes very questionable, right? Because we're always doing simplifications. And if these simplifications actually are important for the physics, then maybe something is fundamentally wrong that we're missing to fix that. So any models that we're using in our simplified theoretical approaches need to be robust, and if they're not, then we have to rethink the models, not the simulations. Also, finite size effects, of course, inevitable. So, you're not running this on an arbitrarily large system size. You need to be aware of that. Usually you're fine, everything's good. But you need to be aware that any finite size restriction is also a finite time restriction. You cannot run the system longer than, once your dynamical excitations reach across your entire system size, that's the last meaningful time step you can look at. Otherwise, you just get periodic recurrences that show up. And if you don't have a periodic system in experiment that you try to model, then you have an artifact, and you just need to be aware of that. Also, effective macroscopic rates are not the microscopic reaction probabilities. They are not that in experiment, they're not that in the simulations. So I was having these kind of microscopic probabilities in my definition of this process. Once I go to a microscopic description in terms of a reaction, say mean field reaction diffusion equation, these are not the same rates. They're always renormalized. Sometimes, I can just kind of refit them and get new numbers, and sometimes this renormalization is a lot more crucial, as I was explaining for these binary annihilation processes. All right, and I think I just skipped the details because we don't have time. So that's an example of Lotka-Volterra, where you have these spontaneous structure formations. This is an example of cyclic Lotka-Volterra, or kind of rock, paper, scissors models, where it depends if you do this kind of realization, which is cyclic Lotka-Volterra, A plus B. No, A it's B, B it's C, C it's A, but just regenerates the particles. So we have total particle number conservation. Or a variant of that where we don't, where we take the A plus B to A predation processes that are cyclic and do the reproduction processes C to C to C, for example, separate. So you split this up. Then there's no conservation law for the total particle number, and that absence of conservation law allows for structure formation. In this case, you can have spontaneous formation of spiral structures. You do not have that in that system. And then thirdly, same kind of nonlinearity. We still have this kind of binary process. Two species come together. One of them gets replaced by the other. That is in all these epidemic models. So SIS and SIR models, the same one. And therefore, too, you have spreading fronts. And as I was saying before, for the SIR model, you get isotopic percolation at the threshold. In all of these cases, there are regions where mean field behavior is violated, and you really need to worry about the correlations. And then finally is this effective macroscopic reaction rates. Maybe I spend 2 minutes on this quickly, if I may. And just on the first example. So let's say we're doing something really simple. We just do a restricted birth process. A goes to 2A. And so we do that with a finite rate sigma. And as we do this initially in a sparse system, very few initial particles, we will always have that reproduction. So it just goes. But then we are imposing a local carrying capacity. Say there's a finite number K of particles we can have locally. And so as we're trying to produce more and more of these, at some point, this carrying capacity will block that actual reproduction process. Right? How would you describe that in mean field theory? By a logistic expression, you would have an effective rate that goes like 1 minus density over K. If you do the simulations, what you can do is you can just measure the macroscopic reaction rates, right? You just let the system go, and per unit time, you just measure the number of reactions happening. Divide out the particle density at that point, and that gives you a time or density dependent reaction rate. That's measured here for the simple process. And near the threshold, so this is as the density approaches the carrying capacity, so that's the large time limit of this process, we find this linear dependence that you get in mean field theory. But in general, you don't. It's a nonlinear function. So the effective reaction rate is not actually captured by the logistic equation. And that's because you have locally different things going on. In some regions, things are blocked. In other regions, you just happily proliferate and keep going. So even for that very simple process, the simplest kind of mean field description doesn't quite work. And likewise in other systems where this effective reaction rate is measurable and quite important and fun to study. And I just refer to that paper. All right. With this, I'm already 5 minutes over with my tutorial. I'd like to stop here and just invite questions.
So I think, in the interest of time, let's just do one question. There's one question from the chat by Yi Jiang. So she's asking, is crowding automatically considered in these models? Do you limit the number of agents that can be on each lattice site?
Yeah, so that is what the local carrying capacity, or kind of this local restriction of your site occupations would do. That would implement that crowding. You don't have to do that. You can make this arbitrarily large and get away with it, but that's how that is implemented. And sometimes it's really important, and sometimes it's not.
Okay, so there's a question from Steffi Jose. Could you explain how diffusive and demographic noise influence the reaction diffusion models, particularly with respect to the coarsening behavior and fixation times?
Ooh, that's
I guess that's a
the last,
longer.
the last question is, lots of open research problems, I think. Diffusion noise, so in the Stoichiometry formalism, it's not in there. In the simulations, of course, it is. Yeah, we're just running the full stochastic process, including everything. Demographic noise is definitely in there because we're looking at an integer number of particles that we are playing with. And so that's completely captured. And the real issue is how far do you have to go to kind of avoid any kind of algorithmic artifacts that I was setting up. So how you describe your computer algorithm. But once you're in sufficiently large situation, in time, not in particle number, you have demographic noise fully captured. In these theoretical approaches, the exact solutions and the Stoichiometry formalism capture the demographic noise. Stoichiometry doesn't really do diffusion noise.